Overdominance and codominance. And here's your source.
You want to start with codominance?
Yeah.
So with codominance,
David, Rucker, Lily,
And Lorena.
okay. Overdominant.
They're gonna
This
start
is
first.
codominant.
codominance is pretty simple. Basically, you've got two proteins that are written in code, and they actually don't necessarily cancel each other out. They just coexist with each other. So they've got a strand that's about equal. So equal that whenever you take your cup of water and your cup of oil right here, mix them together.
So if y'all could do that too, please.
If y'all do that real quick,
You can do the oil, water.
Oh.
either which way, it'll still sit the same.
Now you
Take
see.
that straw out of yours.
Now if you take the straw should demonstrate. Oh.
Yeah, wait. I'll give you $5 too. No,
Really?
so this is our structure that we're working with for the protein, and as you can see, each different protein would be your water and your oil, and they're coexisting around one singular structure.
So, whenever the straw is basically touching both the water and the oil, it's not being, the oil is not overpowering the water is not overpowering the oil. They're both showing up on the straw, which the straw would be like your chromosome, and the two would be your chromosomes. That's codominant.
Interesting.
Alright, so we're going to demonstrate incomplete dominance, which is basically,
Wrap your red in the white.
oh yeah, this one's going to use your red and your white Play-Doh. So this is the one that was in the PowerPoint with like you have a red flower and a white flower and you end up with pink flowers. Well, that's because, and this goes against, well, I was going to like this goes against like the regular ratio, and it happens because like let's say this is your white and this is your red flower. Well, your white protein is like not working, like it doesn't show up. So it's not that you're seeing red and white, you're actually seeing just a little bit of red. It just doesn't function enough to like come up as red against like the white, but it's actually like not showing white. You're just seeing like a little bit of pigment in the red. So
This
it
is
makes it-
due to the partial catalyzation of the pigmentation of each of them. They're not fully being catalyzed. So this gives you the pink color and not just an overall white or an overall red.
add a little bit too
So
much
when
red.
you have two true read alleles and you kind of mix them together, the red is supposed to be dominant over the white, but it's not completely dominant. That's where we get the incomplete dominance. It only shows up a little bit. When
Yeah, that's
So-
what makes me. I should have used a little bit, but I wanted y'all to see it.
It's like this.
It's
Yeah.
turning
Get the
slowly.
pink.
Yeah, it's turning pink.
So your white is still your recessive, so that one doesn't show up at all. But the red isn't completely dominating over the white.
There you go.
So,
We got pink. It's a race.
So if you think about it, the Play-Doh, the compounds of Play-Doh would be the chromosomes, and then the actual color is the phenotype. So that's how it goes through that.
There you go. That's almost pink.
Pink.
It's
Marbles.
marble pink.
We're getting there.
It's
Yeah.
so long.
Now I wanted a perfect pink.
Yeah, so this
Getting
is your
close to pink now.
incomplete dominance.
You're just
What's this pink? Overdominance, yeah.
So, do y'all have any questions about incomplete dominance or codominance so far? Do you understand how both of them kind of work and how they both show up or how they don't show up fully? Okay. Well, this one's overdominance, and basically the different color Play-Doh that y'all have, there's a big Play-Doh and then there's a little Play-Doh. So we're just going to roll this into a ball.
This one's kind of like that, the blood cell, sickle cell demonstration. There's an example that was in the PowerPoint. Just thought like y'all can relate to that too. Which one we were
So y'all can make two balls.
Yeah, just
A
make
Play-Doh?
little balls.
There's gonna be one big ball here, one big one ball.
So when we were talking about overdominance, we used the sickle cell experiment, and basically sickle cell, we're explaining how the wall doesn't fully develop, so your sickle cell doesn't isn't able to grow completely into its full blood cell, it stays small. So that is your small ball would be your sickle cell. It doesn't have to be a complete ball, guys.
Yeah, as long as it takes wood.
And your big one would be your regular blood cell. The way we were going to demonstrate this is your toothpicks. Your toothpicks would act as a virus trying to attack your cell. So when you stick your toothpicks in, there's a lot of viruses attacking your cell. Got some toothpicks. And then when you're sticking it in the sickle cell, not that many.
Yeah, it can only hold so many.
So the virus, since a lot can fit on this one, and your regular blood cell holds more, it's going to be more prone to be attacked and fully taken over.
So therefore, like your small one is the one that becomes like over dominant, and it's actually better to have this one than that one. When you would think that you gotta really attack it.
You gotta. This one would have more, and
so that one wouldn't have enough to.
It wouldn't really survive.
Sickle cell doesn't have the surface area to hold all the viruses and fully let the virus take over. Well, this one, it does let it take over because they're
It's strong enough to support it and stuff. So
yeah.
You really want this one more than that one, and that's what overdominance is.
So this cell burst and your sickle cell would still be alive even after being attacked. Because it wouldn't get fully attacked because it doesn't have as many viruses.
And then even when it does burst, it's not so much to clean up, whereas kind of clean that up.
When this bursts, your virus kind of spreads out everywhere and it's more prone to go all over your body. Versus when the sickle cell burst, it didn't have that many to begin with, so it'd be like less of a cleanup for your immune system too. So that's where your sickle cell is your overdominant over your regular blood cell. Oh. Any questions about overdominance?
Yes. What
And,
is that one? The right one that you had the holding, was that the sickle cell?
That's your sickle cell.
Alright.
That's your underdeveloped cell if you have the sickle cell.
And if y'all go from the PowerPoints that we learned, what would be another form of sickle cell type of disease that y'all would know? Would be malaria? No, it would be malaria. So the sickle cell goes from the malaria because the way it's formed,
Your virus.
and it's not strong.
Malaria.
Yeah.
Yes.
You have the laptop.
I think so.
It should be anyways.
Yeah, it is. The green light's on.
So we're done. Okay.
So let me make some slides. You all did a great job. So give me So let me just add some clarity to you. I like your examples. I want to make sure that we're all on the same page here. So first and foremost, remember we're dealing with a gene here. We're dealing with a gene that eventually is going to be expressed to where it produces a protein. We haven't quite gone into how that occurs, but that's what it's going with. We're dealing with gene sequence. We're not dealing with whole chromosomes. I know you all mentioned whole chromosomes. I didn't want y'all to think that a whole chromosome is doing it. It's just specific genes that are being expressed, and they're regulating this protein that has this factor. Factor 1. Here they're talking about codominance. They're talking about that. There's this protein structure here, which is structure straw. And structure straw that has a phenotype expressed on it. That phenotype is that it's an interaction with oil and water, with molecule, oil, and water. And they can interact equally with molecule, oil, and water. There is no preference over which it interacts with. That's what that codominance basically seems. This equal interaction with this secondary molecule. Again, they did this great. We're showing you the color. Just going back to remind you that it's dealing with a gene. There's a gene that's involved with expression and of a protein. That protein there has to do with having this color because of this lack of expression of that protein. We get the incomplete dominance. Now this one, I love your example. The only thing I want y'all to forget about is sickle cell. The reason I say don't worry about sickle cell, let's look at this in terms of demonstration of, they have a cell A, cell B, that because of a mutation in the gene, cell B is small. This is a normal cell, cell A. Cell A is big, but mutation causes small cells. They have a function. Their function is to help you survive. We'll just say that in general. However, there's a virus that comes in. A virus that needs to attach to the surfaces of the cell. And because cell A, this nice normal cell, more that virus has capabilities of attaching to that cell, increasing its probability of causing disease and killing this particular cell. Cell B, though, because of that first mutation, it isn't as big. It doesn't have as many receptors on its surface, allowing for the virus to attach. Which means that because of that, it has a higher likelihood of survivability during a viral attack. That right there, we refer to as, now this cell is going to be the dominant cell being presented in the blood. This is overdominance. The reason I say that because a sickle cell is not considered a cell that's resistant to malaria. A sickle cell is just another cell that causes problems. Mm-hmm. It is a heterozygote in between. But I like your thing. You just need to have a little bit more clarity. But did you see the distinction there now with overdominance? Using that, it's a great model system. I'll use that in the future.
$200.
They attack.